Hello. Good evening, students. Hello.
Good evening, ma'am.
Good evening. Only you are attending, ma. What about others?
Don't know, ma'am. They're not attending. It seems.
Okay, okay, no problem. So, one second. So yesterday we have discussed about the chromosomal theory of inheritance, and before that I also talked about what is karyogram, what is karyotype, and all those things. And you all know actually to study the chromosomes, we need to prepare a chromosome plate. That the procedure also I have told you how to prepare a chromosome plate. And once you prepare a chromosome plate, you can study the structure of the chromosome in detail and can find out how many chromosomes are present in each fish. Okay, so like that, when once you prepare a chromosome plate, what you need to do is you have to take a photograph, and then you have to cut the chromosomes according to their size as well as the shape and paste it on a paper or a different sheet. So such a phenomena or such a procedure, you call it as nothing but a karyotype. So karyotype actually helps you to study not only the morphology of the fishes, but also it will help you to detect the various chromosomal abnormalities that occurs in the organisms. So that is actually what we call it as nothing but the karyotype. And karyogram is nothing but karyotype or karyogram, it means one and the same. Now karyotype actually is nothing but you um, I mean preparing a plate and then finding out, whereas karyogram actually is nothing but the image of that. So after taking a photograph, you call it as a karyogram. Before you taking that, you call it as nothing but the karyotype. So both mean one and the same. Okay. So here what we can do is, when once you prepare a plate, you have to actually cut those, observe it under the microscope. Then after that, after observing it under the microscope, take a picture of it. After taking a picture, cut them according to their size, according to their shape, and paste it on a paper. Okay, so that we call it as nothing but the karyotype. So karyotype helps us to what? Not only to identify the number of chromosomes that are present in the fishes, and also it will help you to detect the various chromosomal abnormalities. Now, after that, we have also, we know so many actually the fishes have been studied, and their chromosome number also have been given by many of the scientists. And already we have done in the practicals what are the different fishes and what the chromosomal number they have given. The next we have talked about the chromosomal theory of inheritance, both qualitative as well as the quantitative aspects. Now, qualitative means it reflects only the quality wise, like the color of the what do you say, the scales or the color of the body and all those things. Now, quantitative means number of the scales in fishes or number of fins in fishes. So that comes under the nothing but the quantitative trait. So even the the other organisms, including the humans, how the characters are
Excuse me,
inherited
ma'am.
from the one generation to the another generation, or how the characters are inherited from the parent to the offspring. Likewise, in fishes also, the different traits, both phenotypic as well as the genotypic
Ma'am.
traits, is qualitative as well as the quantitative, are inherited from the one fish to another fish. That means from the parent to their offspring. Okay. Now today we are going to talk about the chromosomal manipulation in fishes. Now this is an article actually in which they have given in detail. So that is the reason why I have taken. Now already we know what are the various gene
Ma'am, this is
manipulation.
which chapter?
Hello.
Which chapter it is, ma'am?
This is chromosomal manipulation or genetic manipulation in fishes.
Okay, ma'am.
Genetic manipulation or chromosomal manipulation in fishes. Now, this I have selected because here they have done, this is actually the work done by one of the scientists. So he has mentioned how you do the. I told you know the other day in the class. We have talked about the different manipulation techniques, where how you can actually try to inactivate the gametes by giving the shock treatment, by giving the chemical treatment. Okay, and likewise, and also by giving the various other like androgenesis, gynogenesis, we have talked about. No, the same thing I'm going to deal here. Okay, so this is called as chromosomal manipulation or the genetic chromosomal manipulation or the genetic manipulation in fishes. Okay, so this is a research article and it has been done. So they say that actually the research on the chromosomal manipulation in fishes has a little history when compared to that of what do you say, the vertebrate animals as well as the crops. So since 1943, actually, the earlier attempts were initiated, and until recently, various techniques have been developed to interfere with the normal functioning of the metaphase fetal apparatus during the nuclear cycles of the cell division by using various chemicals. So here also, what we are trying to do, like I mean, after doing the manipulating means doing the genetic manipulation, get is where you may develop the triploids, that is polyploids, and we may also go for androgenesis, gynogenesis, and all those things. Okay, so here by doing the what do you say, the gene manipulation, now the individuals with the differing genomic okay, the polyploids, that is triploids as well as the tetraploids, then gynogenetics, both meiotic as well as the mitotic gynogens, and the androgenics that is are being produced in the fish population. Now, fishes, you all know, actually they are different from other animals, and they are raised in farms. And because for the most of the part of fertilization occurs externally, and the embryological development only takes place without the protection of the internal womb. So whatever the fertilization actually that is taking place, it takes place in the, it is external, it takes place in the water, as you all know. So that is why when compared to the other animals, here it is quite different. So because fertilization occurs externally over here, it is relatively easy to manipulate the chromosome number, they are saying. Because if it is present in the womb, if it is inside the body, it is very, very difficult to go for the gene manipulation. But since the fertilization is external in the fishes, you can manipulate the gene. You can manipulate the gene. That means you can here you can collect the gametes, both the eggs as well as the sperms, that is ova as well as the sperms, and you can manipulate them by inactivating them, by treating them with UV rays or the X-rays, and also by giving the different types of treatment. So a number of techniques actually have been developed to produce the haploid fishes, diploid fishes, as well as the tetraploid fishes, and even to produce fish whose chromosomes actually will be either from their mothers, that is gynogens, or from their fathers, that is androgens. So here gynogens and androgens means if they, I mean, the chromosomes are coming, the whole chromosome or the gene constitution, you can say the composition is coming from the mother, you call it as a gynogen, and if they are coming from the father, you call it as an androgen. Okay, so the techniques that are used to alter the chromosome number are similar, and they are divided into three categories. What is it? One is temperature, pressure, and the chemical shock. Now, most of the researchers actually what they have done is they have used either the pressure or the temperature. Now, these are actually nothing but the physical shocks. So either so by treating them with the hot water, okay, or by treating them with the cold water, that you call it as nothing but the temperature treatment. So by pressure means when you are applying a pressure, then you call it as a pressure treatment. And chemical means when actually you are exposing them to certain chemicals, then it is called as nothing but a chemical treatment. So these physical shocks actually are applied to eggs that are newly fertilized. So the exact timing and the duration of the shock relative to the meiosis and the mitosis actually and the exact temperature or the pressure determine not only the success rate but also it will tell you the type of the chromosomal manipulation. So the pressure chambers actually are used to apply pressure shocks. So the
Ma'am,
device, yes, ma?
ma'am, this is not there in reader.
No, this is there. Chromosome manipulation in what is the genetic manipulation it is. Not chromosomal manipulation. Genetic manipulation in fishes, it is there. You just check the book, gene manipulation or genetic manipulation in fishes. Have you not done in the practicals regarding polyploidy, then androgenesis and gynogenesis? Do you remember? Yes. What, ma?
Yes, ma'am, but you
Uh,
are explaining something different, but it is not there in reader.
no, no. I'm coming to that technique now. I'll tell you. Okay. So this is a basic introduction. So what is actually the treatment they are giving? So physical treatment as well as a chemical treatment. Physical treatment means temperature, pressure. Okay. And the temperature means either the cold treatment you may give or hot treatment you may give, and then you may give you may put the eggs in a pressure chamber and then expose them or apply the pressure shocks. And the other one is a chemical treatment. Okay, so there are several techniques actually that can be used to assess the success of the chromosomal manipulation. What are those? Is one method is to count the chromosomes. Second method is to examine the RBC. Then next is the third method is to examine the erythrocyte nuclear volume. And the fourth method is to determine the DNA content in the erythrocytes. And the fifth method is to stain and count the nucleoli. And the sixth technique actually is to use electrophoresis. So these are actually the various techniques to find out or to assess the success of the chromosomal manipulation. Now this is done after you manipulate the gene and after fertilization, when the result you get it. So that is the newly product or the in the form of fish you get it. That fish, okay, you can take it and you can cut that fish and you can go about with these techniques. So when once the technique is over, you can go for the assessment of the genetic manipulation. So these are the techniques that you need to apply. So before that, what you can do is, what are the various treatments that you can apply is chromosomal manipulation actually can produce triploids, tetraploids, gynogenetic as well as androgenetic. Now, what do you mean by triploid here? So the creation of the triploid is nothing but a type of chromosomal manipulation. Triploids actually are created by shocking newly fertilized eggs to prevent extrusion of the second polar body. So the fertilized egg thus contains an egg haploid nucleus, a sperm haploid nucleus, and a second polar body haploid nucleus, which creates a triploid. So triploids actually have been created in many species. So what they're saying is, when you want to produce a triploid, what you need to give is, you have to collect the egg, okay, a newly fertilized egg, and then what you have to give them the shocking treatment. So means by any shock method, that is by you can give the temperature shock or a chemical shock, or you can give them a pressure shock. Okay, so what happens is, when you are giving this treatment, it will prevent the extrusion of the second polar body. Means the second polar body will not be allowed to form. So what happens then? The fertilized egg actually will contain what? It will have a haploid nucleus, a sperm haploid. So egg haploid nucleus will be there, a sperm haploid nucleus will be there, and a second polar body haploid nucleus will be there. So which creates a diploid. So that means the second polar body will not come out. Okay, it will remain within it. So that is the reason why the union of these three will create a what? It will create a diploid. So diploid. So triploid actually has been created in many species, in so many what do you say, the fishes like Heteropneustes, then Tinopharingo, then Cyprinus carpio, Labio, Clarius. Different species actually the triploids have been created by following this method. Are you understanding? Hello, are you listening, ma? Yes. Hello. Hello.
Yes, ma'am.
Okay. See, this is there in your syllabus. Don't get confused. Okay, gene manipulation in fishes, that is, or genetic manipulation in fishes, or chromosomal manipulation in fishes means one and the same. Okay, so now what is the use of this creation of this triploid fish? They are saying that this triploid fish has a great potential for commercial exploitation in aquaculture. So when you're going for an aquaculture, this actually triploid fish has a great significance. They serve as an attractive model for basic research, and as the triploid fish are with abnormal physiological features, they provide a unique potential for discovery of new biochemical as well as a molecular mechanisms in basic physiology. So future study also needs to be done on these various aspects. Okay, so that is the significance of the triploid fishes. So how do they do now? We have seen it is actually done by giving shock. That shock may be a temperature shock or a pressure shock or a chemical shock or whatever it is. Okay, so this is how you produce the triploid, by manipulating the gene or by manipulating the chromosome. Now next we'll see about the tetraploids. Now, tetraploids actually are what? They are again the fish that have 4 sets of the chromosomes instead of the normal 2 sets. So, tetraploids can be created by what? Again, by shocking a diploid zygote. Okay, you have to take here a diploid zygote. Then, when it is undergoing the first cleavage, then this will prevent what? The nuclear division. So, the nucleus will become 4N and the zygote is tetraploid. So, tetraploid actually is done in Oncorhynchus, then in Tinopharingo, then Labiorohita, Katla, and so many other fishes. Actually, this has been done. So what do you mean by tetraploid? How do they do this tetraploid? Tetraploid actually means nothing but having 4 sets of the chromosomes. So tetraploids again can be created by giving the shock treatment. So either it may be a chemical or a temperature shock or a pressure shock, whatever it is. So what you need to take here, there we have taken the eggs, newly fertilized eggs. Now here we have to take a diploid zygote. If you take a diploid zygote and give it a shock when it is undergoing the first cleavage, what happens? It will prevent the nuclear division. So means it will not allow the further nuclear division to take place. So the nuclear division will be stopped. So what happens? The nucleus will become what then? 4N, and the zygote will become tetraploid. So that is the what do you say, the logic behind this production of the tetraploid. Okay, and the creation of this tetraploid actually is that they are used to produce interploid triploid, so which could eliminate the need to continually create the triploids manually. So tetraploids actually are made to diploids, and the ensuring progeny actually are
Ma'am,
yes ma.
please teach from reader. No, we can't able to understand from this article.
No, I'll come to that later. First, let me finish this. I'll come to that later. Okay. Now here, actually, if you understand the logic, it is very, very simple. So that is the reason why I have selected. Okay, na? Hello. Hello.
Ma'am, but we are not able to understand. We are having reader. We'll read from that if you teach.
Okay. Now, what I'm telling you here is you keep the reader in front of you.
Yes, it is in front of me only.
Yeah, it is in front of you. No. Now turn to page genetic manipulation in fishes.
Yes, I turn.
Yeah.
There are
No.
methods like thermal shock, pressure
Yeah.
shock,
That's
chemical
what.
shock.
They have given different types of what do you say, the this thing. No, first one is I think gamete inactivation, no, or inactivation of the gametes, where what do they do is you can inactivate the gamete by exposing it to UV rays, X-rays, gamma rays, and all those things. Second one, they have given the what is a treatment, shock treatment. Yes or no?
Yes,
Yes.
but no physical shock is there which you are teaching.
That's what I'm telling you here. In that shock treatment only, you see temperature is there, pressure is there, chemical treatment is there. See, cold and hot, if you remember, just go through it.
Okay, yes.
It is there or not?
No cold or no hot. It is there thermal shock, pressure shock, and chemical shock. No
That's
physical
what.
shock.
Okay, actually the thermal shock, pressure shock is nothing but physical shock only. Okay. What you are doing, we are actually physically we are giving no. Okay, we are not giving any. See, what is the difference between physical and chemical shock? When chemical shock is nothing but when you are exposing it to a chemical, then physical shocks come under thermal shock. It may be thermal, it may be pressure, where you are not exposing it to. What you are doing, you are just giving the treatment, means you are putting it in the giving either the hot temperature or the cold temperature, or exposing it to shock, giving the pressure shocks. So that comes under physical mark. Are you
Okay.
understanding? Is it
Okay.
okay? Now it is
And
clear.
yes,
Is it clear?
angiogenesis is there. All the steps are there.
Yeah, that's what I'm coming to that. Okay, I'm coming to that now. So now you got the tetraploid, this thing. So here, why you want to go for the production of tetraploid means here, the main reaction is tetraploid. They are saying that actually it can be used to produce the interploid triploid, triploid. So if you do produce this interploid triploid, there is no need to go for the production of the triploids. They are saying. So tetraploids actually are mated to diploids, and the result you get it is nothing but the what is that, interploid triploids. Okay, so this is about the tetraploids. Then coming to the gynogenesis, I think you have gynogenesis. Now what are gynogens? Gynogens are nothing but these are the fish that are actually having both the sets of the chromosomes that which come from the mother. So gynogens are nothing but the fish. Okay, and in this fish actually both the sets of chromosomes, they come from the mother, they don't come from the father. So the gynogens can be produced for several reasons. The first is to produce a monosex female population, and if the species has a XY sex determining system, then female are XX. So all gynogens will be females. So gynogens actually are produced in one of the two ways. So what do you mean by gynogens? Gynogens are nothing but they are fish. Actually, in here, both the sets of the chromosomes actually come from the mother, and these are actually produced, okay, for several reasons. and they are produced actually to produce monosex female population. Means they don't want the female, they want only female population. So if you want only female population, then you can actually go for the production of gynogens. And if the species has the XY sex determining factor, say for example system, then all the females will be XX. So all the gynogens actually will be females. So there are two methods given to produce these gynogens. One is the eggs are inactivated, so the eggs are activated with sperms which have had their genetic material destroyed, okay, by X-rays, then gamma rays or UV radiation. Shortly after activation, the egg is shocked to prevent the second living. Then the egg now contains two haploid nuclei, the egg nucleus and the second polar body. Now, these are the sets of the chromosomes originated in the mother. So, what they're saying, first, what we have to do, we have to take the eggs, we have to activate these eggs with the sperms, which have actually had been the genetic material destroyed. Means you have to collect the sperms, you have to actually take the sperms whose chromosomes actually are inactivated, you can say, or damaged, you can say, by exposing it to X-rays, gamma rays, or the UV radiation. So here eggs are activated, no doubt. Eggs are good enough, they are active, but the sperms are what? They are inactive. They are inactive means they are destroyed, they are damaged by exposing to them. So such eggs and such sperms, if you allow them to mate, okay, what happens is the egg is shocked to prevent the second living. So here the eggs now contains what? Two haploid the nuclei, the egg nucleus and the second polar body. So these are nothing but the two sets of the chromosomes which are originated from the mother. So here what they mean to say here is, see, you are when you are damaging the sperms, when you are inactivating the sperms, there is no question of the chromosomes coming from the father. Is it okay? So the eggs actually, whatever the eggs are having the chromosomes, that will be from mother only, no? Yes or no? Yes, ma. Hello?
Yes, yes.
So that will be from mother only. So that is the reason why what they are telling is, you have to collect the eggs, you have to collect the sperms. The eggs will be the normal eggs only, but the sperms will be are what we have to actually, before you allow them to fertilize, eggs and sperms to fertilize, that sperms have to be actually destroyed by exposing them to X-rays, gamma rays, or UV rays. So such type of sperms you have to collect, and then you have to mate them with the eggs, means allow them to combine with the eggs. So when once you combine them with the eggs, what happens? Fertilization will take place. No. So the egg will now contains what? It will have only two haploid nuclei. That is, the egg nucleus will be there and the second polar body will be there. So the chromosomes cannot come from the sperms. So whatever the chromosomes that are present in the the egg only will be there. No chromosomes will be coming from the sperm because the sperm is destroyed. The sperm is inactivated. So that is why you can produce the gynogens. That is what they are saying. Second technique is what? The same way. In this, the first step is to activate the eggs with the sperm which have had their genetic material destroyed by irradiation. So this will produce a gynogenetic haploid, same way. But when the haploid zygote undergoes the first cleavage, the embryo is shocked, and this prevents the cell division. The two haploid nuclei has to. So this is also the same thing. Only thing is what they are saying is here the second technique is the same. The same way you have to collect the eggs, you have to collect the sperms which have been already destroyed, and this will produce a gynogenetic haploid. So when the gynogenetic haploid is produced, what you have to do is when this undergoes the first cleavage, the embryo has to be shocked. Shock means you have to give the shock treatment to the embryo. So when you give a shock treatment to the embryo, what happens? It will prevent the cell division. It will not allow the cell division to occur. So what happens? The two haploid nuclei will be there, no? They will fuse to form a 2N zygote. So, and both of the sets of the chromosomes will be coming from the mother only. So there is no question of the chromosomes coming from the father. Okay. So gynogenesis actually is a valuable tool for producing unisex population. Okay. So you can get only unisex or monosex, means only one single population. That is only the from the mother. Okay. That is all the females are produced. Okay. And this particular technique has also been done studied in various species like Catla, Tino, Faringo, and Tino, Faringo, then Cyprinus carpio have been produced by using this technique. Okay. The next is what? Androgens. Now, androgens actually are the opposite of gynogens. Okay. So here we are talking about from the males. So we want actually all the male population. We don't want the female population. So when you don't want the female population, now what you have to do? Now these are the fish actually which are having, these are those fish which are having, which are, means whose chromosomes are coming only from the father. They are not coming from the mother. Like in gynogens, the chromosomes are coming only from the mother, but in androgens, the chromosomes are coming only from the father. So here androgens can be produced by one of these two methods. The most commonly method is to fertilize the eggs whose genetic material has been destroyed by gamma rays or X-rays with normal sperm. So here what you have to do, see in that what we have done, we have taken an active egg and we have taken a destroyed sperm. But here it is a reverse. Are you understanding, ma? Hello? Yes?
No, ma'am. Androgenesis, no.
I'm telling you, see, what is gynogenesis? What is gynogenesis? In gynogenesis, what did I tell you? You have to take a normal egg and sperm, destroyed sperm, no damaged sperm you have to take, I told you, no? Yes or no? Yes?
Yes, yes.
Huh. So in that case, what happens when the sperm is destroyed, when the sperm is damaged, there is no question of the chromosome coming from the father, no?
Yes.
That is why it results in gynogenesis. Now here it is a reverse story. Very simple. Now here what you have to take, you have to take what? The normal sperm, okay, and the eggs, destroyed eggs you have to take. The eggs which have been destroyed by exposing to UV rays, gamma rays, or any other type of rays. Are you understanding?
Yes, yes.
Huh. So what happens here? Now there is no question. The egg will be inactive. No, the egg will become inactive. It is totally damaged. So whatever the chromosomes that are present in the sperms, they will be from the father only. No. Yes
Yes.
or no? So it will result in what? Androgens or gynogens?
Okay.
It will result in androgens.
Androgen.
Did you understand the logic here? Yes or no?
Yes, yes.
Huh? What? Androgens is what? From the father. Gynogens is what? From the mother. So to produce gynogens, what we are doing? We have to take a normal egg and we have to take a damaged sperm and we have to mate them, fuse them. So fertilization will take place. But what happens? But though fertilization takes place, but since the sperm is damaged, the chromosomes will not be passed. Actually the chromosomes will be inherited from the parent to the offspring. So the chromosomes will, here in this case, the chromosomes will not come from the father, but it will come only from the mother. So that is why it results only in the production of gynogens. Whereas in case of androgens, what we are doing? We are actually taking a normal sperm, but we are taking the egg which has been destroyed or damaged by exposing it to different types of rays. Yes or no? So when the mating takes place, fertilization will be taking place. No. So what happens? The zygote will not have the chromosomes from the mother. It will have only from the fathers. Yes or no? Why? Because egg is destroyed. No. Egg is inactive. So the chromosomes from the mother will not be transferred to its offspring. Yes or no?
Yes.
Huh. So that
Ma'am,
is why,
please teach from reader, no?
yeah, that is why I don't have the reader, ma. I'm telling you. Wait. Okay. So that is why actually it results in the production of what? Androgens. Is it okay? Yes or no?
Yes.
Okay. The second technique is the same. To produce androgens is to fertilize the eggs and whose genetic material has been destroyed by irradiation with the sperm from the tetraploid. Okay, the tetraploid males produce the diploid sperm, same way what we have done. Okay, the next is what? Haploid. I'll go to the next one. I'll tell you in detail again. Okay, now till here, did you understand? Yes or no?
Yes.
See, simple logic, because it is very, very simple. You are not understanding the logic over here. Okay. So next is what? Production of haploid. Haploids are what? Individuals that have only a single set of chromosome. They have only a single set of chromosome. So there are two ways to create the haploid, and both involve the destruction of genetic material from one of the parents. So one way is to fertilize eggs with the sperm that have had their genetic material destroyed by X-ray, gamma rays, chemicals, or UV irradiation is probably the best method to destroy. The second way is to create haploids is to destroy the genetic material in eggs. Now this is very, very simple. Now to produce the haploid, what we have to do? What are haploids? Haploids are nothing but the individuals having only one set.
They have
Diploid,
4 set of chromosomes.
yeah. So see, diploid is having 2 sets of chromosomes, triploid having 3 sets of chromosomes, tetraploid having 4 sets of chromosomes, and haploids are having only 1 set of chromosomes. So here, to produce the haploids, what we have to do is, there are 2 ways to create. Okay, and both they involve the destruction of the genetic material from one of the parents. So what we have to do is, one way is, we have to fertilize the eggs with the sperm. So take the eggs, normal eggs, and take what, the eggs which have the sperms which have been destructed, okay, or which have been damaged by the exposing it to the x-rays or gamma rays or the chemicals. Are you understanding? So here, since the what is that one of the parent is damaged or totally destroyed, you will have a single parent chromosome. So that is why it result in only 1 set of chromosome. That is nothing but haploid. So either of the way, you can either damage the egg, or either you can damage or destroy the sperm. So both of them will result. So that means, what happens if the egg is the egg is destroyed, the chromosome will come from the sperm. And if the sperm is destroyed, the chromosome will come from the egg. So that means it has only 1 set of chromosome. No, it doesn't have 2 sets of the chromosome. So there is a question. So that is why it results in only the production of only 1 set of chromosome. That is nothing but the haploid. Okay, haploids are very, very interesting because they enable the breeders to study certain embryological genetic processes. Okay, and practically also, it is very important to know how to produce haploids, because this is the first step in the production of gynogenetic as well as the androgenetic fish. Okay, so this is about, so don't get confused. See the text, and this is why I have chosen this. Is actually, it is very, very simple way. Okay, in simple way to they have done. Now in your syllabus, they have given no gamete inactivation, different methods they have given. Okay, and
Ma'am,
production of
but after androgenesis, gynogenesis, there is diploidization.
uh,
Then, then,
polyploidy is there, no?
then it is application of gynogenesis in aquaculture. Then polyploidy.
Yeah.
There is no haploid, only
Okay.
tetraploid and
Okay,
polyploid.
okay. Let it be, no problem. See, we can see, we can have extra information, no? Yes or no? Okay. So polyploidy is what? Nothing but production of what? Polyploidy is. Huh.
4 haploid gametes.
Yeah, more than two. Generally, it will be diploid. More than two, it comes. So triploids, tetraploids, all will come in polyploid only. Okay, they come in polyploid only. Okay. Is it
Yes.
okay?
Yes. Okay.
Still any doubt now?
No. No.
It is very simple, ma. Just means here they have done it, and they also given the examples in which fish actually they have tried, and they have produced those fishes. Okay. Now, then we'll go to the cryopreservation. One more topic you have, that is cryopreservation of gametes. Okay. Now, what I have talked just now is about the chromosome manipulation in fishes, or the genetic manipulation in fishes. How we manipulate the gene. Okay. To why you are manipulating the gene? Okay. To produce the transgenic fishes. Yes or no? The main purpose behind the chromosomal manipulation is nothing but to produce the transgenic fishes, or to produce the what do you say, the gynogens or the androgens or the polyploid fishes like tetraploids, triploids, and all those things. Is it okay?
Transgenic species chapter is completely different, no?
Yeah, transgenic chapter is completely different.
Okay.
Okay.
You're
Uh,
leaving that chapter and starting this, ma'am.
Yesterday I have talked about the transgenic fish production. I think
Okay.
only one of the girl was present yesterday. I have talked about the transgenic fish production.
Okay. Cryopreservation of gametes.
Yeah, now we will go for cryopreservation of gametes. Is it okay? Any doubt you have?
No. No.
Okay.
Ma'am, please teach from the textbook photos only. That
Okay, okay. Tomorrow I will try to take those because I don't have the book. Okay. Now this is I'll take that. Okay, from sir. I don't have the book, ma.
Okay, because it is easy for us to remember, no, ma'am. That
Okay, okay,
is not there.
okay, okay. Okay, are you able to see now? Cryopreservation.
Yes.
Now the next chapter is cryopreservation of fish gametes. Okay, so in your syllabus you have this chapter. What do you mean by cryopreservation? Now preservation is what? Nothing but preserving. So actually you have the, I think we have talked about the fish seed and all those things, how to collect the fish seeds by using different nets and all those
Yes,
things.
yes.
So
In
when once
practical.
you, yeah, when once you collect those seeds, how to preserve them? Preservation is also very, very important, no? Before you go for spawning and before you go for fertilization and all those things process, that is the production of either it may be a transgenic or whatever it may be. So in that case, what you have to do, you have to preserve them. So how you have to preserve them? Cryo means what? Nothing but at the means thawing. Uh, at low temperature, at very, very, very low temperature, actually you have to preserve them. So cryopreservation actually is nothing but it is the method by which the living cells, okay, or the tissues or the organs, they are preserved and they are viable for an indefinite period, okay, by storing them at minus 196 degrees centigrade. At minus, see the ordinary fridge and all those things, it will have what, 4 degrees centigrade only, but it is in minus. Okay, so actually the cryo preservators will be there where you have actually the what do you say, the low temperature freezers will be there. In that actually you have to preserve this. Okay, so that is what we call cryopreservation. So cryopreservation is what? It is nothing but a technique which involves, means here, suppose you are actually doing any research purpose, okay, and you want actually for an indefinite period of time, you have to preserve them. You cannot preserve them in normal, what do you say, fridge, because normal fridge actually will be having only the temperature 4 degrees centigrade, not more than that. So in that case, actually, it may be for 8 days or 10 days, it may be okay. But after that, actually, it will deteriorate. So that is the reason why if you want to actually preserve them for a longer duration, then you have to actually preserve them in the cold freezer that is called at minus 196 degrees centigrade. Okay, so now cryopreservation of the sperms actually have been done and it has been very, very successful. Okay, so because here what we need to do is actually most of the things, instead of going for the fish eggs, the sperms actually have been cryopreserved. So large size of the egg and the complex structure, presence of several membranes with variable permeability are obstacles in the egg cell preservation. So that is why what they are doing is they are actually going for the cryopreservation of the sperms. To preserve the sperms, it is very, very easy when compared to that of the eggs. So that is why, because eggs are larger in size when compared to that of the sperms, and they have a complex structure and they have a presence of several membranes are there, and that is the reason why it is very difficult to preserve the egg rather than the preserving the sperms. So they are actually, they have been successful in preserving the sperms rather than the egg. Okay, now even fertilization of the normal egg with cryopreserved spermatozoa were not successful. So that is why what they have done is actually they have taken cryopreserved spermatozoa were not successful. So fertilization of the normal egg also was not successful. So therefore, knowledge of the composition of the milk and the sperm quality essential for the optimum rate of fertilization and healthy activity. So instead of that, what they try to do is now fertilization of the normal. See, though we are preserving the sperms, but those cryopreserved sperms, how far they are successful? What they are saying is even the fertilization of the normal egg with cryopreserved also has not been 100% successful. Only to some extent it was successful. So therefore, what they try to do is they try to actually preserve the milk. To preserve the milk. So milk. So what is milk here? Milk is nothing but a fluid actually which oozes out from the male, and it consists of what? Nothing but the sperms. So instead of actually preserving the sperms, what they try to do is they try to preserve the what is the milk. Wilt is what? Nothing but a liquid which comes out, okay, from the male fishes. It comes out from the male fishes. So if you collect that and preserve it, it is more beneficial and it is more successful rather than preserving the sperms. That is what they are saying. Okay. So this is the next is, what are the principles of cryopreservation? What are the steps you have to follow? Now, cryopreservation is nothing but freezing. Just now I have told you, know, so that you have to keep them or put them in the freezers. So there are three essential steps in freezing. What are those? One is called as the freezing. Second one is storing, and the third one is thawing. Now, the rates of freezing and thawing are critical in preserving the vitality and the viability of the spermatozoa. Means how far your what is that? How much time you are taking? I mean, for preservation, and also the duration, the rate of freezing and thawing is also very essential. So all these you have to take. So these three steps you have to follow when you are preserving the sperms. Hello, are you listening? Yes. Hello.
Yes, ma'am.
This is okay? Yes or no?
It is
Yes.
their volume of milk and spermatocrit value, sperm
Uh,
count, sperm motility.
yeah. Okay, so that is afterwards. This is actually with reference to the preservation, means how to preserve them. Okay. Now, so there are three essential steps. There are three essential steps in freezing. They are what? They are freezing, storing, as well as thawing. So what they're saying is, if the cooling rate is very low, it is too low, then the cells will get sufficient time to lose water. It can maintain the osmotic equilibrium with the extracellular solution. So cell death is caused by excessive dehydration. So what they're saying is, if the cooling rate is very low, then what happens? The cells will get sufficient time to lose the water. So it can maintain the osmotic equilibrium. But again, if the cooling rate is very high, then the time for water to escape out of the cell is very little, or no water escapes out of the cell. So that's why what happens? Intracellular freezing will take place and ice crystals are formed. So that is why the cooling rate should be moderate. Are you understanding, ma? Yes. Hello.
Yes, the cooling rate should be at a defined temperature.
Yeah. So it should be at a defined temperature. It should be specific enough. It should not be too low. It should not be too high. So if it is too low, what happens? What happens? It actually, the cell will get sufficient time to lose water, no doubt. Okay, and it can maintain the osmotic equilibrium. But cell death will be caused by excessive dehydration. But if the cooling rate is very high, then there is no time for the water to escape out of the cell. So the water will remain inside the cell only. So intracellular freezing has to take place, wherein ice crystals will be formed. And the cooling rate therefore should be moderate. Only part of the freezable water leaves the water in the cell and forms the crystals which are tolerable. So use of cryopreservation. For that, what you have to do? You have to use certain substances which called as cryoprotectants. Now, what does the use of cryoprotectants? The cryoprotectants actually increase the rate of vitrification and optimize the post-thaw survival. So if you use certain chemicals which are called as protectants, what do? They will actually increase the rate of preservation and also optimize the survival rate. Okay. So what are cryoprotectants? What are the different cryoprotectants that are used? The cryoprotectants are nothing but that they are the chemicals that will minimize the injuries to the cell due to ice formation, or it suppresses the ice formation. So why do we need to use cryoprotectant? What is the main function of the cryoprotectant? These are nothing but the chemicals. Okay. So these chemicals, what do? They will minimize the injuries, okay, to the cell. They will not allow the cell to get damaged, or they will not allow the cell to get destroyed due to the ice formation. Or it will suppress ice formation. So if there is no ice formation, what happens? The cell will be in the life condition, okay, and it will be remain viable for longer duration. So that is why cryopreservants. So it is not just easy just taking out the seam, what is that, collecting the sperms and putting it in the freeze. For that you need to follow certain precautions. So what are the precautions you need to take? You have to actually collect that milled or collect this, the sperms, whatever you are collecting. and you have to freeze them in
Should
the
be
freezers. And
frozen immediately. Should be
yeah,
frozen immediately, and there should be a thermal shock which is given
yeah.
with the ice crystals, and they
Hmm.
should be cooled at 15 degrees Celsius regularly.
Celsius. Yeah. So at minus 96 degrees centigrade, you have to do it. So what happens? And in addition to that, you have to add certain chemical to it. Okay. Use the chemicals. So these chemicals are what? These chemicals are called as nothing but the cryoprotectant. So the cryoprotectant duty, what is the purpose of using the cryoprotectant? It will not allow or it will minimize the damage to the cell due to the ice formation, which occurs due to the ice formation. So it will not allow the ice to be formed. So if the ice is not formed, definitely the cell will remain viable and it will remain live for a longer duration. Okay. Now choose criteria for choosing a cryoprotectant. So what should be a criteria? What things it should have? Actually, it should be least toxic to the cells. So whatever the chemical that you are choosing or you are using as a cryoprotectant, it should be less toxic to the cell. It should be permeable to the cell, and it should be soluble in water during freezing. So these characteristics it should have. So what are the characteristics it should have? One is it should be least toxic to the cell. If it is toxic, what happens? It will kill the cell. It will damage the cell. So it should be least toxic to the cell, and it should be permeable to the cell. Means the cell should be permeable. Means the cell should be allowed, or it has the ability to allow the what do you say, the water as well as the other particles to move from inside to the outside. So it should be permeable enough. That means the cell has the it should have the permeability to make allow the entry of the what is that, the components. Next is it should be soluble in water. So the cryoprotectant also should be soluble in water. Okay.
Ma'am,
Now there, yes,
our fifth semester papers are corrected.
you should not ask now. This is the class going on. No such questions you should not ask. Okay. Later on you can call me. Hello. Is it okay?
Okay. Okay, ma'am.
Okay. Now these are, there are two categories or there are two types of cryoprotectants. One is called as a permeating cryoprotectant. Okay. Permeating cryoprotectant, which are permeable to the cell membrane. Now, permeable to the cell membrane. That means what is it do? What is the function of this permeating cryoprotectant? It will reduce the rate of diffusion of water from cell to extracellular ice crystal. Second, it will reduce the cell volume change and it will also reduce the rate of ice crystal growth. Okay. The other one is a non-permeable. One second. Yeah. Non-permeating cryoprotect. So there are two what do you say, types of cryoprotectants. One is the permeating cryoprotectant, which are permeable to cell membrane. Now, what is the role of this permeability? Means it will reduce the rate of diffusion of water from the cell to the extracellular ice crystals. It also will reduce the cell volume and it will also reduce the rate of ice crystal growth. Now, common the permeating cryoprotectants that are used are DMSO. Now, DMSO means dimethyl sulfoxide. Okay. The full form of DMSO is dimethyl sulfoxide. Second one is glycerol. Then you have methanol, and the fourth one is propylene glycol. So these are the four common permeating cryoprotectants that are used. What are those? One is called as a DMSO. Then you have glycerol, then methanol, then you have propylene glycol. So these are the four methods that are being used. Four cryoprotectants that are used. Okay. Permeating. Now coming to non-permeating cryoprotectants, the non-permeating cryoprotectants actually are not permeable to the cell membrane. Means they do not allow they do not allow the cell membrane to have an entry. Means to it will not means the cell membrane, they'll not allow the cell membrane to become permeable. So if it becomes permeable, actually what happens? There will be access to the entry as well as exit from the cell membrane. But if it is not permeable, the cell will not allow the entry or the exit of any material from inside to the outside. So non-permeating cryoprotectants had got that function. Second thing is it acts by depressing the freezing point and raising the ice formation temperature of extracellular solution. So what it will do, it no doubt it will decrease the freezing point, but it will raise the ice formation temperature. The temperature of the ice formation, if it is raised, of the especially of the extracellular solution. So these are the 2 points. And the commonly used chemicals, that is the cryoprotectants, are what? Sucrose, glucose, then you have dextran, egg yolk serum, skim milk, as well as the antifreeze protein. So these are actually the different types of non-permeating cryoprotectants. So we have seen what is a cryoprotectant. It is nothing but a chemical which is actually used for the cryopreservation purpose. There are two types of cryoprotectants. One is permeating cryoprotectant and the other one is non-permeating cryoprotectant. And we have seen what are the different examples. Okay. Now, apart from this, also you can add extenders. Now, what are extenders? Extenders are nothing but
Ma'am,
solution of balanced salts.
ma'am,
Okay.
we are not having in this brief about cryopreservants.
You're not having.
This much briefly about cryopreservants. Only like one paragraph is there for cryopreservants, like types and not non-permitting. It is not there.
Acha, types are not there. Okay. What is cryopreservation is there. That's all, no?
Yes.
Okay. Okay, no problem. Now, what are extenders? Extenders are nothing but solution of balanced salts. Okay. So, apart from these cryoprotectants, what you have to do is you have to actually also prepare a balanced salt solution and you have to mix them and add. So, actually, it is used to dilute the milk, as undiluted milk is not suitable for the freezing. So, why you use these extenders is actually we are saying that we have to store or we have to preserve the milk. What is milk? Just now I have told you, it is nothing but actually the liquid that oozes out from the what is it, the males, which contains what? Which contains the sperms. So this extenders, what do? The extenders actually dilute the milk. They dilute the milk, because if you dilute the milk only, it is suitable for what is the preservation. If you don't dilute the milk, what happens in the undiluted state? You cannot freeze them. So that is why if you dilute the milk, okay, it will be suitable for the freezing. Okay. Then also it will inhibit the activation of spermatozoa and functions as a medium for cryoprotectant. So second thing is what? It will also inactivate, you can say, inactivate the spermatozoa. That is, inhibit the activation of the spermatozoa. Okay. Now, the diluent used for milk is combination of extender and cryoprotectant. So, you have to keep it in the fridge as it is an exothermic reaction. Three categories of diluents you have to use. That is extender A, how much quantity you have to take. If you want, you can just read it, and if you don't want, you can omit. So, here what they have given is, now extender A 90 ml, DMSO 5 ml, glycerol 10 ml. So, you have to make a solution of this and you have to use. Then extender B 90 ml, DMSO 8 ml, glycerol 10 ml. Extender C 65, DMSO 15 ml. So any one of the diluent, you can mix it
Ma'am,
with the milk. Huh.
what is thawing?
I'll tell you, I'll come to that. Okay. After freezing, you have to, the next process is nothing but thawing. I'll tell you. Okay. Now freezing is what? Keeping it in a freezer. Okay. To, what is it? So that it will not
Storing
get,
at
yeah, it will not
storing
get
at minus degrees Celsius so that the sperms don't get damaged, and the
damaged. Yeah.
cryopreservants are used.
Yeah. Yeah.
Protected.
Yeah. So now that the diluent used for milk is combination of extender as well as a cryoprotectant. I told you, no, these are the three categories you can use. Any one of these diluents, you can mix it with the milk in a proportion of 4:1, and you can keep it in the freezer. So they have given how much of DMSO you have to use, how much of glycerol, how much of, you cannot just take it as you like it. Okay. So the certain proportion is there, and if you do it accordingly, then you will be successful. Otherwise, it will get damaged. So these things you need to take. Now, what are the advantages of cryopreservation? Now, coming to the cryopreservation advantages. Now, in seasonal breeders, okay, such as carp seed can be produced and supplied to farmers at any time of the year. Second thing is it helps in selective breeding and in development of other techniques meant for genetic upgradation. Then it helps in preventing inbreeding depression. It helpful in preserving genetic diversity and in conserving the endangered species. Reduces the cost of brood stock maintenance, and it is also useful in conditions where male and female mature at different times. So these are the advantages of cryopreservation. So if you preserve them, you have got a ready-made. It will be, so whenever you want, you can go for it. So that is why they are saying that in seasonal breeding, see, breeding will not take place throughout the year, no. So in seasonal breeding, the seeds can be produced and supplied to the farmer. So if you actually have got the preserved seeds in the form of the sperms or in the form of egg. Of course, eggs cannot be preserved. Just now we have talked only about the sperms. So if you are preserving the sperms at any time, actually you can go for the breeding. Yes. 1 second. One second, ma. Okay. So all this we have just now finished. Advantages. So these are the different advantages we have seen. So cryopreservation is very, very important. We have to preserve them so that we can breed, okay, and supply the seed at any time. And also, it is useful in condition where male and female they mature at different time slots. So that is why it is very important. And we can also reduce the cost of the brood stock maintenance, and it also prevent the inbreeding depression. So these are the different advantages. Now next, coming to now here one example they have given cryopreservation of calf milk protocol, how they have done, how we can do about. Okay, so just now I have already told you about the different the same thing, how to prevent example of a calf milk. So healthy male breeders actually are selected during peak breeding season. Then they are subjected to hypophysation. Then within 3 hours, milk from the males is collected. Then yield of the milk is so and so. Care is taken to avoid contamination of milk. Then milk is stored in the fridge, that is in a freezer. The sperm count and the sperm motility is assessed. Then if more than 80% of the spermatozoa are motile, okay, then the milk is diluted by a mixture of extender and cryoprotectant. Then 3 types of extender solutions are used. Then they are stored in a refrigerator for 1 month. Diluents are prepared by mixing. Okay, all this already have done. So this is the procedure they have given. This mixture is kept and then mixture. Okay. Then whenever required, the reactivation of the sperms can be done by thawing. Fast thawing is preferred. Over slow thawing. So French straws are made to slush by swirling them in a water bath at 38 to 70 few drops. Now actually just now you have talked about thawing, no? Now see whenever actually you are freezing it. Now in ordinary fridge, what we do when we keep ice or anything like that, okay, and we take out the ice from the fridge, no? Yes or no? Yes, ma?
Yes, ma'am.
So in that case, what we do